Welcome to G Fun Facts Online. Today, we are stepping into a world that surrounds us every day. A world we have walked through, eaten from, and built our lives upon, yet have never truly understood. We are talking about the plant kingdom. But specifically, we are talking about the end of silence. For the entirety of human history, we have viewed plants as the passive backdrop to animal life. We see them as scenery that breathes but does not speak. We assumed that because they lack a mouth, they lack a voice. We assumed that because they lack a brain in the way we understand it, they lack thoughts or intentions. We were wrong. Beneath the rustling leaves, beneath the quiet expansion of roots in the soil, a complex, high-speed exchange of information has been occurring for hundreds of millions of years. It is a dialogue of survival, a chatter of warning, and a negotiation for resources. Until now, this language was encoded in a biological cipher so complex that it appeared to be mere molecular noise. But that silence is breaking. We stand at the precipice of a new era in biological science, driven by the convergence of two distinct intelligences. The first is the ancient, adaptive intelligence of Flora. The second is the modern, computational, pattern-recognizing intelligence of artificial intelligence. Today we are discussing the development of the Plant RNA Decoder, specifically the breakthrough model known as Plant RNA-FM. This marks the moment we stopped just looking at plants and started listening to them. This is not science fiction. It is the dawn of a reality where we can upload the genetic code of a crop and receive a status report, a warning, or a request for help. To understand the magnitude of this technology and to fill the next 20 minutes with the density of information you expect from this channel, we have to start at the very bottom. We have to look at the alphabet of life itself. To understand what the AI is decoding, we first must understand the medium of the message. Biology has long operated under what we call the central dogma. This is the flow of information in biological systems. It goes like this. DNA makes RNA, and RNA makes protein. For a long time, the public focus has been almost entirely on DNA. DNA is the blueprint. It is the static library of information stored in the nucleus of the cell. It is the hard drive. If you sequence the DNA of a plant, you know what that plant is, you know its potential, but DNA is static. It doesn't change from minute to minute. If a plant is thirsty, its DNA looks the same. If a plant is being attacked by a beetle, its DNA looks the same. So looking at DNA doesn't tell you what the plant is experiencing right now. At the other end of the chain, you have proteins. Proteins are the bricks and mortar. They are the physical structures and the enzymes that do the work. But the middle step, RNA, or ribonucleic acid, is the true architect. RNA is the messenger. It is dynamic. The population of RNA molecules in a cell changes constantly based on stress, the time of day, the temperature, and who is attacking it. If you want to know what a plant is, you look at its DNA, but if you want to know what a plant is feeling, thinking, or doing, you must look at its RNA. For decades, scientists knew this. They knew that RNA was the language of cellular decision-making. But there was a massive problem. The language is incredibly difficult to read. It is written in a 4-letter alphabet of nucleotides: A, U, G, and C. These are strung together into sequences that can be billions of letters long. But unlike a sentence in a book that reads from left to right, RNA is three-dimensional. It folds, it twists, it knots itself into complex shapes. A letter at position 1 might be functionally connected to a letter at position 500 because the molecule folds over and touches itself. To a human researcher, a long string of RNA looks like chaos. It looks like random noise. We could see the letters, but we couldn't understand the grammar. We couldn't predict how the shape determined the function. This is where the second intelligence comes in, artificial intelligence, specifically the technology behind large language models, or LLMs. We are all familiar with models like ChatGPT. These systems are trained on billions of words of human text. They learn that the word apple is statistically likely to be followed by the word pie or tree. They learn syntax, grammar, and context. They don't just memorize definitions. They understand the probability of the next token in a sequence. Researchers at the John Innes Center and the University of Exeter asked a radical question that changed everything. They asked, what if we trained a large language model not on English or Chinese or Python code, but on plant RNA? This led to the creation of Plant RNA-FM. The team, led by Professor Yiliang Ding and Dr. K. Li, compiled a staggering dataset. They didn't just take a few samples. They fed the AI 54 billion sequences of RNA from 1,124 different plant species. This training corpus included everything from ancient mosses to modern commercial crops like wheat and maize. They fed this massive library into the neural network. And just as a human LLM learns that a verb usually follows a noun, the Plant RNA model began to learn the grammar of genetics. It started to recognize patterns that no human eye could ever see. It learned that a specific sequence of nucleotides usually folds into a hairpin loop structure. It learned that a certain pattern in the 5' untranslated region of the strand means make a lot of this protein urgently. It deciphered the syntax of life. This is the black box of biology being cracked open. The AI treats RNA structures as words and the full sequence as a sentence. It can predict structure, which is how the molecule folds in 3D space. It can predict function, which is what the RNA will actually do inside the cell. And it can predict regulation, or how efficient the plant will be at turning this message into protein. So we have the translator. Now the question is, what are the plants saying? This is where the science becomes incredibly practical and, frankly, startling. The messages are flooding in, and they generally fall into 3 categories: distress, defense, and adaptation. Let's start with distress. Consider the concept of thirst. Traditionally, a farmer knows a plant is thirsty when it wilts. The leaves droop, the color changes. But by the time a plant physically wilts, the damage is already done. Cellular growth has stopped, and yield has been lost. The plant is suffering. With the RNA decoder, the timeline changes. We can sample a leaf and sequence its RNA. The AI analyzes the transcriptome, which is the collection of all RNA messages currently active. The AI identifies a specific surge in RNA molecules associated with osmotic stress. It sees the plant is talking about water conservation. It sees orders being sent to close the stomata, the tiny pores on the leaf surface, and to shift energy toward root growth to find moisture. The AI translates this massive dataset into a simple English sentence. The plant is experiencing microdrought stress. Crucially, it tells us this 48 hours before any physical symptoms appear. This is the difference between reactive farming and proactive communication. The second category of message is defense. Plants are under constant siege from fungi, bacteria, and insects. When a fungus attacks a wheat crop, it tries to hijack the plant's cellular machinery. The plant responds by producing defense RNAs. These are like tactical orders sent to the cellular army. Before this AI, we might see that a plant was sick, but we wouldn't necessarily know exactly what was attacking it until we saw the fungus itself. But the AI can read the specific grammar of the defense response. It can distinguish between a fungal attack, a bacterial infection, or an insect infestation. The plant uses different words for different enemies. When a caterpillar bites a tomato leaf, the plant releases specific volatile organic compounds and synthesizes specific toxins. The RNA profile for biting insects is distinct from the RNA profile for a mosaic virus. This allows for a level of precision agriculture we have never seen. Instead of blanketing a field with a broad-spectrum pesticide that kills everything, a farmer could receive a notification that says, "Sector 4 is under attack by aphids." The response can be surgical. The third message category is adaptation, specifically regarding heat. As climate change warms the planet, plants are constantly adjusting their internal chemistry to survive heat spikes. The RNA decoder reveals the heat shock response. This is a flurry of molecular messages ordering the production of protective chaperone proteins. These proteins literally hold other proteins together so they don't melt or unravel in the heat. By reading this, scientists can identify which crop varieties are genetically calm under heat and which are panicking. This allows us to breed climate-resilient supercrops much faster than before. To truly appreciate how the AI does this, we need to look under the hood of the technology for a moment. We need to talk about the transformer architecture. This is the same neural network structure behind GPT-4. Transformers are unique because they utilize a mechanism called attention. In a human sentence like, "The bank of the river was flooded," the AI knows the word bank refers to land, not a financial institution, because it pays attention to the context words river and flooded. It looks at the whole sentence at once. Biology works the same way. In an RNA strand, the function of a nucleotide at position 1 might depend entirely on a nucleotide at position 500. Old AI models read sequences linearly, from left to right, and they missed these long-distance connections. They couldn't see how the tail of the molecule affected the head. The transformer architecture sees the whole molecule at once. It understands the complex 3D relationships that define biological function. It solves the folding problem. Knowing the sequence of letters is easy. Knowing how they knot and twist into a functional machine is hard. Plant RNA excels here. It identifies complex structures like G quadruplexes. These are knots of 4 guanine bases that act as on-off switches for genes. By predicting these structures with high accuracy, the AI allows us to see the machinery of the plant's language, not just the text. This brings us to the application of this technology, which scientists are calling Agricultural Revolution 2.0. We are moving from the Green Revolution, which was based on fertilizers and basic genetics, to the Smart Revolution, based on information and communication. The most immediate application is smart breeding. Traditional breeding is slow. You cross two plants, you wait a season, you harvest the seeds, you plant them, and you see if the offspring is better. It takes years. With the RNA decoder, breeders can analyze the seedlings weeks after germination. They can ask the AI, does this seedling have the RNA grammar for high yield? The AI analyzes the regulatory elements in the RNA and predicts the plant's future performance. This cuts breeding cycles from years to months. But let's look further ahead. Imagine the concept of the sentinel plant. Imagine a massive field of corn. Scattered throughout this field are sentinel plants, genetically identical to the crop, but equipped with microsensors, or perhaps sampled regularly by automated drones. These plants act as the voice of the field. Sentinel-1 sends a message. My nitrogen-sensing RNA motifs are active. Feed us. Sentinel-2 sends a message. I detect viral RNA fragments. Pathogen alert in the northeast corner. Sentinel-3 reports. My circadian RNA clock is out of sync. Heat stress is disrupting sleep cycles. The farmer receives these updates on a dashboard. The language of Flora becomes a push notification. The farm becomes a conversation. Perhaps the most futuristic application is the development of RNA sprays. This is the new pesticide. If we understand the language, we can speak back. Scientists are developing sprays containing specific RNA molecules designed to silence a pest's genes. For example, if a beetle eats the leaf, it ingests an RNA designed by AI to specifically turn off the beetle's appetite gene. The beetle stops eating and dies. The RNA degrades into the soil harmlessly within days. It is not a toxic chemical. It doesn't poison the groundwater. And crucially, because the AI designs the RNA with such specificity, it only affects that one species of beetle. It is gibberish to the plant, it is gibberish to bees, and it is gibberish to humans. It is a sniper rifle instead of a carpet bomb. Beyond agriculture, this technology offers a new lens for ecological listening. We have heard of the Wood Wide Web, the mycorrhizal fungal networks that connect trees in a forest. We know trees share resources, but we haven't understood the negotiation. Using portable RNA sequencers and AI, ecologists could tap into this network. We could listen to an old-growth forest. We could ask, is the forest stressed? Is it preparing for a masted year? A massive release of seeds? This could revolutionize forestry management. Instead of guessing which trees to log or protect, we could ask the forest which nodes are supporting the network and which are independent. We can also use this for biodiversity monitoring. We use environmental DNA, or eDNA, to find animals in water. Environmental RNA, or eRNA, is the next frontier. Because RNA degrades quickly, finding it means the organism was there recently and was biologically active. AI models could analyze air or water samples to give a real-time status update of an ecosystem's health, decoding the collective RNA chatter of thousands of species simultaneously. Finally, we must consider the future capability, programming nature. If we can read the code, can we write it? The ultimate goal of the Plant RNA Decoder is not just translation, but authorship. Professor Ding and his colleagues have noted that this breakthrough opens the door to synthetic biology. We could design RNA sequences that don't exist in nature. Imagine a drought-tolerant RNA switch. The AI generates a sequence that folds into a sensor for cellular water levels. When water drops, the structure unfolds, exposing a genetic code that triggers immediate root deepening. We are moving toward programmable plants, crops that can be updated like software to deal with changing climates. Of course, this brings us to the ethical frontier. With this power comes profound responsibility. Speaking the language of plants means we can manipulate them more deeply than ever before. There are risks. Could a programmed RNA message jump species? If we design a signal to make wheat grow faster, could it accidentally tell a weed to grow faster too? And there is a philosophical question. Does decoding the language of plants change their moral status? If we know a plant is screaming in distress, even if it is just a chemical scream, does it change how we feel about harvesting it? The AI gives us the power of a god over the plant kingdom. We must ensure we use it with the wisdom of a gardener. In conclusion, the Plant RNA Decoder is more than a tool. It is a bridge. For the entirety of human history, we have looked at the green world across a chasm of silence. We guessed at its needs, we marveled at its resilience, but we never truly understood its internal life. The convergence of artificial intelligence and plant biology has built that bridge. The Plant RNA FM model and its successors are the translators of a conversation that has been ongoing since life moved onto land. We are learning that the quiet world is not quiet at all. It is a cacophony of molecular messages, complex, urgent, and sophisticated. By using AI to speak the language of Flora, we are not just improving crop yields or fighting climate change. We are acknowledging that we are not the only intelligent entities on this planet. We are finally learning to listen. And the plants? They have been waiting to tell us everything. Thank you for listening to G Fun Facts Online.